My name is Rafael Hernandez, and I've been asked by my co-chair today to introduce our session with a discussion of the serologic and immune responses to SARS-CoV-2, both in general and, well, the limited amount we know about individuals with cystic fibrosis. I have no disclosures relevant to today's presentation, other than the fact that this is a rapidly changing field, and it's possible that what I present today may be out of date compared to when I recorded it a month ago. But I do want this to be a jumping off point for our discussion during this workshop. The objectives of my talk today include initially just briefly reviewing SARS-CoV-2's viral structure, so we're all starting on the same page, discussing the mechanisms of, in general terms, of SARS-CoV-2 immunity, and then in more detail discussing the antibody or serologic responses to SARS-CoV-2 infection, both after infection and vaccination, how the antibodies correlate with protection, and possible impact of CF on antibody responses. So first, SARS-CoV-2. It is a single-stranded RNA beta coronavirus. And these are called coronaviruses because they resemble a crown, as you can see on this electron microscope here. And these protrusions are, in fact, you can see them better in this 3D model of the coronavirus, the spike protein, which protrudes from the surface of the virion. The spike protein is notable for several reasons. First, the distal portion of the spike protein actually binds the ACE2 molecule on human cells, which is a receptor, and initiates the infection process. Second, the spike protein is the target for currently approved or emergency authorized vaccines in the United States and Europe. There are multiple other proteins that are important in the structure of SARS-CoV-2, and the one that we will talk about briefly today is nucleocapsid, which is internal to this structure and bound to the RNA genome, and it's also a strong immunogen. First, a brief overview of what we think of as the natural immune response to respiratory and other viral infections in general. And this is what we think occurs in mild cases of SARS-CoV-2. After initial exposure, the virus begins to replicate and increases in number. And this triggers an innate immune response very early on during the infection. This has several effects. First, it's the initial breaks that slow down the initial viral replication within the cells. And second, it recruits adaptive immune response cells, such as T cells and B cells, that will produce a specific response against SARS-CoV-2, eventually bring down the viral load significantly so that symptoms resolve and the infection clears. However, in severe cases of SARS-CoV-2, a different picture has emerged. It appears that after initial exposure, the virus begins to replicate and significantly increase in numbers, and this initial innate immune response is delayed. This allows the virus to reproduce to high levels, but also delays the initial adaptive immune response. And then when both of these arms of immunity kick in, they seem to kick into higher levels than those observed in mild cases. And this causes excess inflammation and more severe symptoms, leading to things such as requirement for ICU level care in severe cases of COVID. So although today we're going to focus primarily on the antibody responses in the adaptive immune response, I do want to point out that the innate immune response has also been implicated as being important in determining outcomes after SARS-CoV-2 infection. And this is one example. Defective type 1 interferon signaling has been associated with severe COVID-19 disease. And so these are two sister studies that looked at defects in type 1 interferon signaling that seem to contribute to life-threatening, or at least correlate to life-threatening SARS-CoV-2 infections. So they looked at a population of asymptomatic or mild cases of COVID-19 versus those with life-threatening cases. And they looked for genetic differences in proteins in the interferon signaling pathways. And the reason they chose these genes and this type 1 interferon pathway is it had been previously implicated in being important to prevent severe pneumonia after influenza infection. And what they found is that more than 3% of patients who had life-threatening SARS-CoV-2 infections had loss of function in these signaling pathways due to genetic differences, as opposed to none in the mild or asymptomatic cases. And in a complementary study, they also looked at acquired defects in interferon signaling. And here they were looking for antibodies that are able to block interferon signaling. And in patients with mild or asymptomatic disease, they saw that they didn't generate any antibodies that blocked their own interferon signaling. Whereas in those with life-threatening disease, they saw that 10% of patients had antibodies blocking interferon signaling. Again, supporting the idea that interferon signaling is important for gaining control of SARS-CoV-2 early during infection. But we're going to focus the majority of the remainder of the talk on antibody responses to SARS-CoV-2. And there are a lot of studies that have looked at this, but this is one good study that illustrates the timing of antibody responses after natural infection. And it's from a cohort of adults in Tucson, Arizona, where they looked at antibody responses to multiple antigens, including the receptor binding domain of spike protein, the S2 domain of spike protein, and nucleocapsid. And these are all for IgG, but the timing for IgM is very similar. And what you see is that the antibody responses quickly come up during the first 7 to 10 days after symptom onset, and that by 14 days, almost all patients have detectable antibodies to each of these antigens. And also, most patients have detectable neutralizing antibodies that are able to prevent infection by SARS-CoV-2 on cells in culture. In addition, when they looked at durability of these antibody responses, they followed a subset of these patients and additional patients from a community cohort over periods of time up to 6 months after symptom onset. And what they saw was after that initial increase in antibody levels to each of these antigens, they slowly decline, but that antibodies to spike protein in 2 different regions, both the receptor binding domain and the S2 domain, persist. Whereas for nucleocapsid, you see a similar trend, but by 6 months, some patients have the level of antibody has fallen below the detection limit. So we see that natural infection induces antibody responses that last for up to 6 months. What do we see in vaccination? So this has been reported for each of the vaccines that's been tested in clinical trials. And this is just one example from the mRNA 1273 trial, which is the Moderna mRNA vaccine. And the reason I chose this is it was the data following antibody responses 6 months after vaccination has recently been released. So here you see these antibody levels. On the top, this is for antibodies to the receptor binding domain. And on the bottom, it's neutralization antibodies using a live virus assay. And this is the time frame from vaccination. So the 2 arrows indicate where the first and the second dose of vaccine were given, and they go out to 6 months after the initial vaccination. And what you can see is vaccine, both receptor binding domain antibodies and neutralizing antibodies increase after the first dose, but then increase markedly after the second dose, and then gradually decline over time, but are still detectable in all of the small group that was initially in this phase 1 trial. Notably, antibody levels are slightly decreasing faster in adults who are over age 55, but they do persist in all participants. So we know that natural infection and vaccination can induce neutralizing antibodies, but these actually correlate with protection. And there's multiple studies that have come out during the latter part of the summer that suggest that they do. And one of the more comprehensive ones is one that modeled data from 7 individual vaccine trials looking at levels of antibody and vaccine effectiveness. And so as a visual display of what they did, essentially here you can see that if you measure the antibody levels in participants in the vaccine, some vaccines induced high levels of neutralizing antibodies, while others, such as the COVAX vaccine, had lower levels of antibodies. And then if you compare this to vaccine effectiveness, where maybe only less than 10% seem to be susceptible to disease versus 50%, you can come up with an estimate for the antibody level that seems to be protective. And because the different vaccine trials use different methodologies to measure antibodies, they normalize the antibody levels to the antibodies that are seen after natural infection or convalescent antibody levels. And so if you plot the normalized antibody levels versus the vaccine effectiveness, you can see you can basically fit a curve to that with the uncertainty for each of these points estimated by the hash. And then to assess how functional their model was at predicting vaccine effectiveness, they then did a leave out analysis where they rebuilt the model with 6 of the 7 trials and saw how well it predicted the efficacy of each vaccine that had been left out with multiple iterations. So the observed efficacy of a vaccine versus the model's ability to actually predict the efficacy of the vaccine. If it were perfect, they would all sit along this line. But you can see that the model actually predicts most of the efficacy for these vaccines relatively closely as they're lying along relatively near the line here. So since we can see that SARS-CoV-2 induces strong antibody responses, and these antibody responses seem to correlate with protection against infection, what do we know about the pathophysiology of CF that might infer that serologic immune responses might be different? Well, they may not be, but there are some things that suggest that they are. And that's one, that chronic infection can cause dysregulated inflammation. And so the chronic lung infections observed in CF could actually contribute to differences in immune responses. Second, historically, IgG levels in many people with CF were either higher or lower than normal expected for age. But recent studies have suggested that these are actually closer to the typical levels of immunoglobulin observed in people without CF. And this is probably due to improved disease status regarding better lung health and nutritional status. But there's also been low seroconversion rates reported occasionally for vaccines, such as pneumococcal polysaccharide vaccines or H1 influenza vaccine. But these reports are rare, and it's tough to make any generalizable statements. There is limited data on the immune responses to SARS-CoV-2 in people living with CF. And so I'm going to present here some very preliminary data from a study that we're conducting in Seattle. And so there's more information on this study on poster 25 from our fantastic pulmonary fellow, Georgine Hergenroder, on both information on our cohort and clinical symptoms. But in brief, we recruited 125 children and adolescents from CF clinic at Seattle Children's and followed them for 12 months with intermittent serology and weekly symptom and exposure surveys. All seroconversions that we observed were mild or had no symptoms, and this study is still ongoing. Only a small fraction of patients have actually completed their 12-month serology testing and all of their symptom surveys. But we do see some trends emerging that I think are worth discussing. And the first is that spike IgG levels are higher after vaccination versus natural infection. And so you can see that here, where the spike IgG levels are quite a bit higher compared to patients who had infection but have not been vaccinated. Second is that the immunoglobulin spike IgG levels seem to persist for quite a while after vaccination. And so most of these patients received the Pfizer mRNA vaccine because most of them were under 18, but you can see that vaccine-induced antibody levels are still quite prominent months after their vaccine doses. So are there other factors that may contribute to differences in immune responses in patients living with CF? And one of those things that we should keep in mind is lung transplantation. In general, solid organ transplantation and the immunosuppression that's required for transplant is known to affect vaccine responses. And in particular, lung transplant patients require higher levels of immune suppression. And following COVID vaccine, multiple groups have described that vaccine responses are quite low in patients who have undergone lung transplant. So in this cohort in Texas, only 25% of patients who had a lung transplant mounted a measurable or positive antibody response to spike protein after either Pfizer or Moderna vaccines. And even those who did have positive vaccine responses were on the lower end compared to non-transplant controls. It's worth noting that patients may still develop T-cell responses even if they don't have antibody responses. So is there anything we can do to help these patients develop better immune responses? And yes, we can provide a third dose of vaccination. So one study that supports this is a randomized controlled trial looking at a third dose of mRNA 1273 vaccine in all types of solid organ transplant. And it's been shown to induce higher levels of spike immunoglobulin, even across individual patients. You can see pairwise here, higher neutralizing antibody titers, and also higher levels of polyfunctional T cells active against SARS-CoV-2. So in summary, SARS-CoV-2 immunity is mediated by innate immune mechanisms, as we described briefly, B cells and antibodies, and T cells, which help to coordinate the antibody response, but and also have other cellular control mechanisms that we didn't discuss today. Neutralizing antibody responses correlate with protection from symptomatic COVID-19 after both natural infection and immunization, but we need more information on immune responses to SARS-CoV-2 in people living with CF. There's many outstanding questions, such as how long immune protection will last after infection or vaccination, what vaccination schedules will prevent severe or symptomatic infections over time, and whether or not CF disease status influences COVID-19 immunity, and hopefully we'll be able to answer those in the future. I'd like to thank the organizers for an opportunity to speak, all of the fellow workshop speakers today who are contributing to this session, and the scientists whose work I included in this overview. I'd also like to thank the team at Seattle Children's who has contributed over the last year to the preliminary data that I presented today. If you have any questions, I'd be happy to answer them at this time.
So I'm Rafael Hernandez, who just presented that initial invited speech talk here at the request of my co-chair, Raymond Pickles, who's working behind the scene to help field questions for our session today. I would encourage all of you to go ahead and put any of your questions into the chat box. And if you are interested in the same question that somebody else has, you also have the opportunity, of course, to either like or vote that up higher in the priority chain. So it looks like we have one question. The first question today is whether or not there's any data to suggest that CF carriers have any protective advantage against SARS-CoV-2 relative to the general non-CF population. And I will say that at this point, I am not aware of any data that supports that supposition. And in fact, I think at this point, the quality of data suggesting the actual risk for infection amongst people who have CF is also not entirely clear, since all the reports in persons with CF are all based on case reporting through registries, and so it's based on testing as opposed to actual infection rates. But what has become apparent is it doesn't, at least with the data presented so far, that most patients with CF are not at any obvious greater risk for disease. It looks like we have another question. Are there any data to suggest immunosuppression may increase susceptibility to infection, yet decrease the risk of cytokine storm? So there has been some suggestion of that in the literature, and I know that colleagues who work with other immunosuppressed populations, either solid organ transplant and the hematopoietic stem cell transplant populations, are actively collecting and evaluating that data. I am not aware, and certainly some of those patients do seem to be at increased risk for after infection with SARS-CoV-2, but there may also be some signal that some certain circumstances may reduce the risk. And so I am not aware of any definitive data one way or the other at this point. Another question, will there be any T-cell data in people with CF at some point? It seems that an important data point for the community to understand. I assume that, and this is from Amy Silvis, and I assume that question relates to T cells after either vaccination or infection. I think that one of the difficulties with T cell data is that although there has been a proliferation of reports describing T cell responses in the general population, we don't have very much data on even the antibody responses in persons living with CF, much less T cell data at this point. And so it's going to be, I think right now we don't have a great understanding entirely of how the T cells are contributing to protection in the general population, although there's been a good description of kind of the breadth and variety of T cell responses. We know even less in CF at this point, and so I think it's going to be difficult to know. What has been described is the fact that in patients who have had solid organ transplant, that antibody and T cell responses don't always correspond, at least the way that people are able to measure them. And so there are patients who have measurable and significant T cell responses after solid organ transplantation and vaccination who don't have measurable vaccine responses, and vice versa. But I think understanding how much T cells are contributing separate from B cells to protection from SARS-CoV-2 is challenging, because the large studies that really give us the data to understand correlates of protection at this point are the large vaccine clinical trials, and we don't have as much detailed T cell information from those. And in the smaller, earlier phase trials or smaller studies, we don't have the signal necessarily to call out protected versus not protected. Let's see, if we don't have any more questions, I did want to call out some data that, the limited data that we do know about antibody responses in SARS-CoV-2. There are several posters that are, so first there was one publication that came out relatively early in the pandemic related to seroprevalence in the first spring of 2020 in Belgium, and they showed that there was a 2% seroprevalence for SARS-CoV-2 antibodies in the CF Center in Brussels. But there are multiple posters at this study that are starting to explore this, and one is poster 58 from Kathleen Mahan from the University of Minnesota CF Center, and they describe a 3% seroprevalence rate within their first 6 months of testing, and that to date, their overall seroprevalence rate is 13.2% and highest in the age group 12 to 30 years of age. As I mentioned, our study at Seattle Children's, we've seen a 6.4% seropositive rate to date in our overall clinic population, which really is up to age 20. And that, as I said, was in poster 25. Poster 490, Anna Blanchard at CHU St. Justine and the University of Montreal has initial results from the CAR-CF study arm, which is a study looking at seroprevalence and symptoms associated with COVID-19 in Canada, the US, and Europe. She has initial results from the Canadian arm of that, which they're just ramping up testing, but looking at that poster, it's obvious that there'll be a lot more useful data coming out of that. It doesn't look like there's any further questions at this point, in which case I would say we can go ahead and move on to the next talk, which is going to be from Loic Guillot, and he's presenting Pseudomonas aeruginosa modulates SARS-CoV-2 infectivity in CF airway epithelial cells by increasing expression of the host protease TMPR SS2.
Good afternoon, everybody. I would like first to thank the organizer to have selected our abstract in this workshop about SARS-CoV-2 and COVID-19 in cystic fibrosis. I am Loic Guillot. I'm coming from Sorbonne Université in Saint Paris, France. I have no relationship to disclose related to this presentation. So our work is a basic science work in which we study the role of Pseudomonas aeruginosa infection in the context of SARS-CoV-2 infection in CF airway epithelial cells. So today, various national and multinational clinical studies of COVID-19 have been performed in cystic fibrosis, and these studies have been reviewed in this journal, and so far the clinical of SARS-CoV-2 in people with CF is quite similar to the general population. However, some patients at higher risk, such as post-transplant, and also patients with lower lung function. And so it's very important to understand why this patient at higher risk. And so we decided to perform some basic science experiment to understand what happened. So during the course of CF lung disease, patients are colonized infected by bacterial pathogens such as Pseudomonas aeruginosa, but also virus, including influenza, rhinovirus, and respiratory syncytial virus. These infections are successive or in the same time, and it's co-infections. And they have an impact in the immune host response, and of course, modify the clinical severity of the lung disease. And what we would like to understand is this complex interplay between bacterial pathogen and respiratory virus. And of course, it's also important with SARS-CoV-2. So we hypothesize that Pseudomonas aeruginosa is able to modify SARS-CoV-2 cellular response. And what is known between the interplay between SARS-CoV-2 and airway epithelium is that SARS-CoV-2 infects the airway epithelial through a receptor called ACE2, and also action of a host protease called TMPRSS2, which is able to cleave the protein at the surface of the virus called spike. Entry of the virus is also associated with an activation of signaling pathways leading to cytokines, chemokines, and a lot of mediators that are able to activate the innate and adaptive immunity. The first thing we try to understand is if Pseudomonas aeruginosa was able to regulate molecule crucial for virus entry in the cells. So we looked at ACE2 furin and TMPRSS2 in CF and non-CF primary bronchial epithelial cells infected with Pseudomonas aeruginosa. So this data from a previous transcriptomic study, here you have the fold change, and you could see easily that TMPRSS2 expression is highly induced in CF cells. And there's no effect in furin and ACE2. If you look at this well detailed here, you could see that there's quite a variability in CF patients. We also confirm this induction of TMPRSS2 using a virulence factor of Pseudomonas, the flagellin. And as you can see, similarly to what we got with the live bacteria, we see that TMPRSS2 mRNA is induced both in non-CF and CF airway epithelial cells. In contrast, as we have seen with the live bacteria, flagellin was not able to modulate ACE2 expression. So TMPRSS2 is induced by Pseudomonas aeruginosa and flagella. To go further and try to understand what happened, why this TMPRSS2 is induced, we use cell line. And firstly, we test expression of ACE2 and TMPRSS2 in KLU3, BIS2B, and 16HB cell lines. And we have seen that KLU3 are expressing a high level of ACE2 and TMPRSS2 in comparison to the other cell line. If you look at ACE2 receptor, it's only expressed in KLU3 cell line. So we further work with these cells. In particular, we use isogenic KLU3 wild type and knockdown for CFTR obtained from Marc Chanson in Geneva, and these cell lines were obtained by CRISPR-Cas9 technology. So as we performed before with primary cell, we stimulate the cells with flagellin 3 hours or 6 hours, and we have seen induction of TMPRSS2 both in wild type and KD cells, even if a strong induction was observed in CF cells compared to the wild type. We confirm this induction at the mRNA level, at the protein level. Indeed, when we stimulate cells with flagellin, we see an increase of TMPRSS2 expression, whereas nothing happened for ACE2. So Pseudomonas aeruginosa is able also to induce TMPRSS2 in CFTR deficient KLU3 cells. To go further, we try to dissect what are the signaling pathway involved in this induction, and we look at the receptor of flagellin, toll-like receptor 5, the MAP kinase P38, and the transcription factor NF-kappa B. These two molecules were previously shown to be involved in flagellin signaling. So as expected, when we use an anti-TLR5 antibody, we are able to prevent this induction of TMPRSS2, and we obtain the similar result when we use chemical inhibitors of P38 and NF-kappa B molecule. We see that we are not able to induce TMPRSS2, and we verify that P38 and NF-kappa B are activated when the cells are incubated with flagellin. And when a P38 inhibitor is used, NF-kappa B activation is prevented because P38 is upstream NF-kappa B. So here we show that flagellin-induced TMPRSS2 expression in CFTR deficient cells is TLR5, P38, and NF-kappa B dependent. Of course, the next question was, is this SARS-CoV-2 infectivity modified when TMPRSS2 is increased? So we pre-incubated KL3 cell polarized at the liquid interface with flagellin overnight, and then infected them with SARS-CoV-2. And then we performed virus quantification. And as expected, we have seen that in cell deficient for CFTR, there is more virus, which was expected because there was a little more TMPRSS2. And this infection is also increased when we pre-incubate cells with flagellin. However, more surprisingly, when we look in the apical media to see particular release, we've seen that in cells deficient for CFTR, less virus. This suggests that CF cells are more able to fight against virus. So in the first conclusion, we could say that Pseudomonas aeruginosa and flagellin increase TMPRSS2 and modulate SARS-CoV-2 infectivity, and this increase is P38 dependent. And this is interesting because a paper in Cell last year showed that P38 pathway is a major pathway related to infection by SARS-CoV-2. And they have done this study by phosphoproteomics. It's a very nice study if you're interested. The limitation of our study is the use of the Cal3 cell line that may not recapitulate the complete epithelium differentiated. So we use another model to see if there's a specific signature of CF bronchial epitheliums infected with SARS-CoV-2. So we use 3D reconstituted epithelial cells from CF and wild-type donors, infecting them with the virus 24 hours, 48 hours, and 72 hours. And we look at epithelial integrity and inflammation by multiplex ELISA. Very interestingly, when we infect cells with the virus, we have a loss of epithelial integrity, which was expected and it's already known. But surprisingly, when we use CF cells, they have an integrity lower at baseline, but there's not so much effect of the virus. So these cells seems protected. When we look at inflammation, at 24 and 48 hours, we didn't see any difference when the cells were infected or not in control group or CF group. At 72 hours, we've seen induction of inflammation with RANTES, IL-17A, interferon gamma, IL-1 beta, TNF alpha, and IL-8 release. But one of the most cytokine or chemokine induced was IP10. And IP10 is very interesting because it is involved in leukocyte trafficking. It modulates innate adaptive immune response, cell growth, and enterostasis. And it has been shown to be a severity marker of COVID-19. And it's also a potential biomarker of CF because it has been shown to be increased in acute exacerbation. And also flagellin is known to induce IP10. Also, it's a risk factor for chronic lung allograft dysfunction, so it may be important for lung transplant. And very recently, it has been shown that low IP10 has been detected in CF patient, and in a CF patient with COVID-19, it's only one patient with mild disease. The level of IP10 is very low. So this IP10 could be a marker of CF, maybe in CF patients. So to conclude for this part, in comparison to control bronchial cells, epithelial integrity is not so affected. No inflammatory response in CF bronchial cells infected by SARS-CoV-2. And so the CF bronchial epithelium may be protected, and IP10 also could be a marker of severity. So to conclude, we've seen that Pseudomonas and flagellin increase TMPRSS2 and modulate SARS-CoV-2 infectivity. The CF bronchial epithelium seems protected. And thus, what we want to know now if IP10 could be a marker of a COVID-19 disease in CF. So I would like to thank our lab, Cystic Fibrosis, Physiopathology and Phenogenomic, led by Ariette Corvor, Manon Ruffin, Jeanne Migaud, Claire Camel, and Viviane Bawa involved in this project. Olivier Terrier, our collaborator in Lyon, and also Sorbonne University that funds this work. And thank you a lot for your attention.
I would like to thank Loic for contributing his talk. Unfortunately, he was unable to join us today for the live discussion, but I would like to point out that the staff from the CF Foundation has put in the chat for this session on the Slido. If you click to the chat, there should be contact information for Loic should you want to get a hold of him and ask him your questions. And with that, we'll move on to our next talk, which is the role of IL-13 induced Th2 inflammation in host responses to SARS-CoV-2 from Shreya Ghimer.
Hello, everyone. My name is Shreya Gimire. I'm a third-year graduate student in Interdisciplinary Graduate Program in Informatics in the University of Iowa. So the title of my talk today is the role of IL-13 and IL-17 induced inflammation in airway epithelial responses to SARS-CoV-2. There is no relationships to disclose regarding this presentation. So inflamed lungs respond to challenges abnormally. For example, here I'm showing you healthy airway. So when healthy airway gets exposed to respiratory viruses, it results in transient inflammation followed by resolution. In contrast, in sick airway, which I'm showing you here by a layer of mucus caused by chronic inflammation, and chronic inflammation can be oversimplified into two general patterns. Namely eosinophilic or IL-13 high, which is seen in most asthma, some COPD, or neutrophilic IL-17 high, which is seen in CF, some COPD, and infection. And when this chronic inflammation gets exposed to respiratory viruses, it results in acute exacerbation, further lung damage, and inflammation persists. So from here, we can see that chronic inflammation causes abnormal responses to viral infections. But does this affect COVID-19? From a clinical standpoint, comorbidities can affect COVID-19 outcomes. And as we know, COVID-19 outcomes vary. It can go anywhere from people being asymptomatic to organ failure and death. And how COVID-19 outcomes is affected by comorbidities is also it's different. For example, older age, obesity, heart, liver, immune diseases lead to severe type of COVID-19 outcomes. Whereas there are some data that suggest that people with CF, asthma, COPD might show less severe outcome of COVID-19, whereas there are some other papers that show the reverse. So clinical and epidemiology data on lung diseases and COVID-19 outcomes is conflicting. But how does inflammation affect epithelial responses to SARS-CoV-2? So hypothesized that IL-13 and IL-17 induced inflammation affect airway epithelial responses to SARS-CoV-2 infection in vitro. So going over my experimental overview, we had cells from human airway epithelia, 3 donors grown in air-liquid interface that were treated with either vehicle, IL-13, or IL-17, and cytokines were pre-treated at 2 different time points, namely early and late. And then the cells were infected with SARS-CoV-2 MOI.1 for time course no virus, 6 hours post-infection, and 72 hours post-infection. And then for this total sample of 48 total samples and 15 condition, we did combinatorial single-cell RNA sequencing using split-seq. So split-seq is a combinatorial indexing strategy done at the bench, which requires no additional custom instrument, and we found the method is compatible with BSL-3 work at low cost. For additional description on this method, you can find it in this website. So now, since we were using this method for the first time, our first stage of analysis involved finding that if this method accurately detects effect of cytokines in human airway epithelia. So here what I'm showing you is a dot plot where y-axis are the treatment groups, vehicle, IL-13, and IL-17 TNF-alpha, and x-axis are the top genes differentiating treatment groups. For example, here we see IL-13 high genes that we kind of expected to be here compared to our standard 10x Illumina data. And then we also saw the IL-17 high genes. And this assured us that this data can actually accurately detect the effect of cytokines in human airway epithelium. And here, the dot represents the percentage of cells that express this gene, and color represents expression level. And then now here, what I'm showing you is a UMAP of cell types or labeled and colored by vehicle, IL-13, and IL-17 TNF-alpha treatment groups. Here, each dot represents a cell, and similar cells are grouped together. So here, these basal cells express the So gene markers such as TP63, KRT5, and they clustered together. So in conclusion, combinatorial single-cell RNA-seq was able to correctly identify expected cell types and detect upregulation of expected genes by IL-13 and IL-17. Now, we wanted to know, do cytokines affect viral titers in vitro? So here, what I'm showing you is a plaque assay of viral titers. Here, x-axis shows the different cytokine treatment groups. Here, for example, I'm just showing you the vehicle. And in y-axis, we show the plaque-forming unit per ml. So here, at first, when we treated the cells with cytokines, for a short period of time, we didn't see any effect. But when we treated the cells with cytokine for a longer time point, we see a massive difference between IL-13 and IL-17 TNF alpha. So it proves that it just shows that IL-13 protects human epithelia from virus in vitro. Next, we wanted to see how it affects viral tropism. So in here, in x-axis, we have 3 different treatment groups, and y-axis shows the percentage of cell types. And here, I've further divided the cell types percent into infected and non-infected cell types, denoted by plus and minus. So for example, here, we can see almost all the secretory cells are infected in vehicle. Very less cells are infected in IL-13, and in IL-17, there are more compared to IL-13 that are infected. Next, we see almost all the ciliated cells in vehicle that are infected, which is kind of expected because there are so many data that suggest or tell that ciliated cells are the first one to get infected. But with IL-13, we see the infection of ciliated cell decreases, but in IL-17, it kind of remains same as vehicle. Surprisingly enough, all the basal cells were also found to be infected in vehicle, and almost all of them were infected in IL-17 TNF alpha, but only half were infected in IL-13. So overall, we see all the common cell types were affected, and IL-13 decreases viral transcript detection in all common cell types. But how? So here are the potential models for IL-13 mediated protection. So IL-13 induced inflammation may block the viral cells from binding to the cell, or might block from entering inside the cell. So if either of these two things happen, then cells are less likely to get infected, but once infected, might show the normal viral transcript levels. The next, the IL-13 induced inflammation may block the replication. If this happens, then we can find decreased virus in infected cells. And such as there might be others possible mechanism. Now I'm going to show you a rather complex heat map, because so heat map in here, we have SARS-CoV-2 related genes and receptors such as ACE2 and TMPRSS2. So coming over here, for example, I'm just showing you the vehicle group here. So each column represents a condition or a treatment group. For example, this is vehicle, time point. And then the first row is control, where cells are not treated with cytokine or virus. But here the 72 represents the cells in this vehicle group were treated with virus for 72 hours post-infection. And then the cells that are treated with virus can further divided into infected versus bystander, meaning even though the cells were exposed to virus, these were the cells that were infected, but these were not. And then the scale just shows the abundance of this gene in each different group. So I'm not going to go into details in this graph, but I'm going to talk specifically about these two groups, vehicle infected, and then IL-13 long treatment group infected. So here I'm showing you a violin plot where x-axis is vehicle and IL-13 long treatment group for 72 hours post-infection. Y-axis shows the expression level, and this S gene is one of the SARS-CoV-2 related genes. So looking at this graph, we see that the levels are kind of comparable between vehicle and IL-13. Again, here each dot represents a cell. Now we wanted to know if epithelial antiviral response increased due to infection. So here again, I'm showing you the same kind of graph, but this time with interference-stimulated genes. Again, not going into details, but focusing especially in these two groups, vehicle infected, and then IL-13 long treatment group infected. Here I'm showing you one of the ISG or interference-stimulated gene, OAS1, which was interestingly found to be associated with SARS-CoV-2 virus recently. So in here, again, comparing these two, we can see that IL-13, whether or not the IL-13 treatment, or whether or not the cells are exposed to IL-13 doesn't matter because it kind of looks same. So from this, we can conclude that IL-13 treated airway epithelia are protected from SARS-CoV-2. However, when an IL-13 treated cell is infected, viral load and ISGs are normal. So this data suggests that IL-13 induced inflammation decreases viral binding and entry rather than replication. So then here, what I'm showing you is a heat map of single-cell RNA-seq data of IL-13 time course. So this epithelium were treated with IL-13 for 0 to 7 and 21 days. So looking at this 21 days, or like late time point, we see the main differentially expressed genes are goblet cell genes, and most of them have antiviral activity. So we came with a possible mechanism that when the cells are treated with IL-13, it induces goblet cell metaplasia, which is denoted here by a thick layer of mucus. And then when the virus tries to infect the cells, the mucus blocks the viral infection by blocking the binding of viral to the cells or the entry of the cells. So in summary, IL-13 and IL-17 TNF alpha affected abundance of viral receptor in all cell types. However, only IL-13 affected in vitro susceptibility to SARS-CoV-2. The protein secreted by IL-13 induced goblet cell may protect epithelia. And again, the effect of cytokines may be different in vivo than in vitro, considering the immune system of in vivo. So lastly, I want to acknowledge Zulu Lab, Dr. Alejandro Zulu, my mentor, Lab mates, Paul McRae and Stanley from my lab, University of Iowa Cell Culture Core, Charlie Rocco for Spitzik Bio, which is now Pars Bio, and also all the funding that we received. Thank you.
Shreya, thank you for your fantastic talk. I'll go ahead and start with a question, which is, it seems that your model suggests that it's this goblet cell hyperplasia and increased production in antiviral genes and also potentially mucus that's reducing the potential infection from SARS-CoV-2 in your epithelial cells after IL-13 exposure. Have you guys gone on to try and test any of those things directly, as far as, for instance, overexpressing any of those antiviral genes, or trying to stimulate mucin production through other means?
Thank you for the question. Yes, the data that I showed today was mostly focused on in vitro stuff. We are in the process of doing in vivo stuff, but it's still in the process, so we don't have enough data to come to any conclusion. But yeah, it's possible that any other mechanism can also induce goblet cell metaplasia and hence protect from SARS-CoV-2.
And then it looks like we have a question that asks whether simply washing the mucus off after IL-13 exposure can abrogate or increase the infectivity again.
That again is a great question, and definitely something to look into. We haven't done that yet, but yes, we can treat with IL-13, wash the mucus off, and then try to see if it still protects the SARS-CoV-2. But yeah, we can't say that from the data that I've shown today, but that's definitely something we could do and figure out.
Another question related to the goblet cell hyperplasia, is the hyperplasia that you're seeing in your single cell RNA-seq? Is that supported by histopathology, or have you guys looked at all?
Yes, we have seen in vivo as well that IL-13 increases goblet cell metaplasia. I haven't shown you data in this, but yes, we have data that shows IL-13 increases goblet cell metaplasia in both in vitro and in vivo.
And then kind of a speculative question, I guess, that I have, thinking about whether or not it's the actual role of the increased mucus production that is contributing to the increased infectivity for SARS-CoV-2 after IL-13. Do you think, because of that, do you think that highly effective modulator therapy such as ETI and cystic fibrosis that might decrease the mucus burden, would that have a paradoxical effect on the risk for SARS-CoV-2 infection if in fact there's less of a mucus barrier overlying those cells in the airway?
That is an interesting question. And it's kind of difficult to say because there are data out there which suggest that people with asthma were not affected with SARS-CoV-2 or were like less infected or were protected somehow from the infection. It might be same in cystic fibrosis, but there isn't enough data to come to any conclusion on that.
And then a question from Daniel Cook. Is there any evidence of increased COVID infection in individuals who are on IL-13 blockade by dupilumab? So an IL-13, so they're on an IL-13 blocking antibody. Is there any evidence of increased COVID infection in those patients that you're aware of?
No, as I shown, the viral titers are kind of same in untreated and IL-13 treated cells. But as I showed, there are less cells that are affected with SARS-CoV-2 with the IL-13 treatment. So yeah, we don't, I guess it does, but you have to do more experiments to figure that out.
And then a question from Camille Ayer. Does IL-13 treatment affect ciliated cells? Are genes involved in viral entry, replication, or egress affected in the ciliated cells?
Yes, we've seen genes, ciliated cells were one of the top cells that were highly affected by SARS-CoV-2. And then we do see that in IL-13, although IL-13 protects ciliated cells, but there are still more ciliated cells affected, even treated with IL-13. So, yeah.
All right. Thank you for answering our questions. It looks like we have answered all the questions from the audience at this point in time. And so we will go ahead and move on to our next speaker, who's Elizabeth Menton, who's going to describe the impact of elexacaftor, tezacaftor, ivacaftor therapy on pulmonary exacerbation rates during the COVID-19 pandemic.
Hello, my name is Elizabeth Menton. I'm a first-year medical student at the NYU Grossman School of Medicine in New York City. Prior to matriculating at NYU, I worked as a clinical research coordinator at Columbia University's Adult Cystic Fibrosis and Lung Disease program, and I'm here today to share some of the research that came out of my time as a member of this team. Specifically, I will be speaking about the impact of elexacaftor, tezacaftor, ivacaftor, ETI therapy use on pulmonary exacerbation rates during the COVID-19 pandemic. I have no actual or potential conflicts of interest to disclose in relation to this presentation today. As many of us are aware, the FDA approved ETI therapy for use within the CF population in October of 2019. This therapy was widely anticipated to improve CF disease prognosis. Clinical trials had demonstrated significant improvements in a variety of clinical measures ranging from percent predicted FEV1 to improvements in pulmonary exacerbation rates and BMI. 2020 represented the first full calendar year since approval of this new therapy. And of course, given this, there's widespread interest in understanding the impact of this ETI therapy on a population level. However, as many of us are also aware, March 2020 saw the arrival of the COVID-19 pandemic to the United States. There was a significant decrease in healthcare utilization rates observed within the general population, as well as the CF population, which was widely considered to be at high risk for this disease. Some researchers have estimated about 41% of United States adults to have avoided medical care in the medical setting due to concerns related to the COVID-19 pandemic. I have included an interesting graphic here, just demonstrating the percent change in outpatient visits per week from baseline over both the course of 2020 and a representative reporting year. So on the x-axis, we have the weeks of the year, 0 to 52, and on the y-axis, we have the percent change in outpatient visits per week. And in dark blue, we see the 2020 trend, which is significantly below the typical year trend, especially between weeks 11 and weeks 24 of 2020. And while there is a rebound observed, this rate still remains below the typical year trend, as we can see in the latter portion of the year. So we have two concurrent phenomena of interest. We have avoidance of the healthcare setting on a large population level, both within the United States as a whole and also within the population of patients followed at the adult CF program at Columbia University. At the same time, we have the introduction of this highly effective triple combination therapy to this patient population. So the question then becomes, how can we untangle the effect of healthcare avoidance on reported exacerbation rates and understand how effective this newly approved ETI therapy is in terms of measures of interest, specifically pulmonary exacerbation rates. So putting it all together, the question then becomes, can any observed decrease in the documented pulmonary exacerbations in 2020 at a single adult CF center, specifically Columbia's adult CF center, can this decrease be independently attributed to ETI therapy within the context of the decreases in reported exacerbation rates over the course of the COVID-19 pandemic? So how can we begin to answer this question? Well, I went ahead and collected data from the CF Foundation Patient Registry, hoping to make a comparison of exacerbation rates between the patient population of patients taking ETI therapy and those not taking ETI therapy. So I've included some helpful definitions here that were used to synthesize my data. Treatment populations were defined as follows. The ETI population included patients who started ETI therapy within the first quarter of 2020, and the non-ETI population included patients taking modulator therapies other than ETI over the course of the year. Exacerbation types were also broken down into two main categories. We have mild, moderate exacerbations, which were defined as those requiring oral antibiotics, as well as severe exacerbations, which were defined as those requiring IV antibiotics and/or hospitalization. Patients with lung transplants, as well as those enrolled in CFTR modulator studies, were excluded from this analysis. So then how can we go ahead and quantify and develop a measure that is useful in assessing the difference between these two populations, the population of patients taking this highly effective triple combination therapy and the patient population not taking this therapy? So we used a measure called, which we called the exacerbation incidence rate, or the EIR. which can be generally defined as the number of exacerbations per group normalized by the number of patients within that group. For example, in this case, the exacerbation incidence rate of severe exacerbations within the ETI population, or the patient population taking triple combination therapy, can be defined as the number of severe exacerbations recorded within this ETI population, normalized by the number of patients within this ETI population. So the results of this analysis begin here, where we do confirm that there was a observable and significant decrease in the exacerbation incidence rates between 2019 and 2020. So this just demonstrates, that there was, as expected, a decrease, whether that's attributable to the COVID-19 pandemic and avoidance of healthcare settings, and thus a reduction in the reported exacerbations, as well as, the potential for triple combination therapy to reduce these rates. And we have here a graphic that demonstrates this. So on the x-axis, we have the various exacerbation types. On the y-axis, we have the exacerbation incidence rate. And this is for series between 2019 and 2020. So we see that there is a significant decrease across all exacerbation types between 2019 and 2020, that there is something going on here that we're interested in evaluating and kind of untangling the different effects of the COVID-19 pandemic and healthcare avoidance versus triple combination therapy impact on these populations. Diving in a little deeper to kind of digest this more and understand what's really going on, we can look at exacerbation incidence rates stratified by both exacerbation type and the treatment population in 2020. And we see that there is a significant reduction across all exacerbation types within the ETI population. So on the x-axis, we have the exacerbation type between mild, moderate, severe, and all exacerbations. And on the y-axis, we have the exacerbation incidence rate. And we see between the three population groupings, the ETI population, the non-ETI population, and the total population, that there's a significant difference between the ETI and non-ETI population. So we can see that there is something going on here that distinguishes the ETI population from the non-ETI population. And we're interested in evaluating whether this between group difference can be attributed to the fact that the ETI population consists of patients taking this new and highly effective triple combination therapy. So diving in a little bit deeper again to go ahead and assess this between group difference and the impact of triple combination therapy, we have a graphic that describes the percent decrease in exacerbation incidence rates between 2019 and 2020, stratified by exacerbation type and treatment population. So again, we have the various exacerbation types grouped along the x-axis and the percent decrease in exacerbation incidence rates along the y-axis. And we can see that the decrease in exacerbation incidence rates was much more pronounced within the ETI population when moving from 2019 to 2020. And this is observed across all exacerbation types. So what does this mean? Of course, all patients were affected to some degree by healthcare avoidance over the course of the COVID-19 pandemic. But the exacerbation incidence rate decrease within the ETI population is much greater than that within the non-ETI population, indicating that ETI therapy has a significant positive impact on pulmonary exacerbation rates and bringing these down over the course of 2020. So just to recap and synthesize some of those big takeaways, between 2019 and 2020, there was a greater reduction in the overall exacerbation incidence rate within the ETI population as compared to in the non-ETI population. And what this means is that ETI therapy had a significant impact in reducing exacerbation rates within the population of patients taking this medication. Additionally, greater reductions in severe exacerbation incidence rates as compared to mild moderate exacerbation incidence rates were observed in both populations, both patients taking ETI therapy and those not taking ETI therapy. However, this reduction was much more pronounced within the ETI population. That is, the severe exacerbation incidence rate was much more reduced within the ETI population as compared to the non-ETI population. And importantly, the between group differences between the ETI population and the non-ETI population were found to be statistically significant across all exacerbation types. So our conclusions from this analysis is that there is a statistically significant difference between the overall exacerbation incidence rate for the ETI population and that of the non-ETI population over the course of 2020. Significantly, the overall exacerbation incidence rate within the ETI population was less than 30% of that of the overall exacerbation incidence rate within the non-ETI population. So ETI therapy really had a significant impact in reducing those pulmonary exacerbation rates, even when taking healthcare avoidance and decreases in reported exacerbations into account. So we conclude that highly effective ETI therapy likely contributed to a considerable decrease in documented exacerbations beyond that attributable to the COVID-19 pandemic alone. I would like to thank Dr. Emily Domingo and Dr. Claire Keating, the directors of the Adult CF and Lung Disease Program at Columbia University, whose support and guidance was crucial to this project and to putting it all together. I would also like to thank the CF Foundation and the Patient Registry Program, which maintains such an incredibly valuable research resource in the patient registry. I would also like to thank my colleague, Turnjot Soroya, for her help in formulating and editing this presentation and its associated materials. And I have just included some references here. Thank you very much for your time.
Thank you, Elizabeth, for that excellent talk. It looks like our first question is from Noel Meltzer, who's asking, can you parse out the decrease in pulmonary exacerbations noted for ETI in the phase 3 clinical trials from the decrease specifically for infection with COVID-19?
Sorry, so we didn't include data from patients who had been enrolled specifically in modulator trials. Did I understand that correctly?
Maybe let me rephrase the question. I think that the maybe what they're getting at is that in the phase 3 clinical trials, you may have seen that patients who started ETI had a decrease in exacerbation rate after starting it. So there's data that's separate from the data that then you collected during from your clinical cohort and your study center. How do those rates compare to each other? And does that give you some sense? Is your rate comparable to the decrease in exacerbations maybe that was seen during the phase 3 clinical trials?
You know, that's actually a great question. I don't have an answer to that, but that's definitely something to look into and draw a comparison to. I think you know, the context of kind of my investigation was more interested on the clinical side and kind of accounting for many of the factors that aren't controlled for within you know, a study. evaluating that more. But yeah, thank you for that question. It's definitely something to look into and a good comparison to make.
And then another question, is there any data identifying the causes of exacerbations between 2019 and 2020?
So exacerbations were strictly pulmonary exacerbations. So in terms of causes, we're looking at respiratory infections in terms of bacterial associations and things like that. We were looking on a larger scale than that. That data would be available and of course would be interesting to kind of dive in deeper and say, on an organism level, are we seeing less exacerbations from one or the other? But I don't have that information for you, but thank you for that question.
And then a question that I had, overall, it does suggest that your data from your center suggests that a large amount of the decrease that we see in exacerbation rates during the COVID pandemic is likely a result of patients starting ETI therapy during that time frame. But it also looks like the patients in your non-ETI group had some decrease in their exacerbation rates. It is a little bit of speculation, but do you think that's associated with the infection prevention controls in the community that were implemented in the community, decreases in respiratory viral rates? How are you guys interpreting or thinking about that decrease in the non-ETI group?
Yeah, I would definitely say that some of the factors that you pointed out were contributors. As many of us are aware, those kinds of infection control procedures were somewhat commonplace within the CF community even prior to COVID. But I think the extent of social distancing and precautionary measures that people were taking at large would be contributing to that decrease that we're seeing within the non-ETI population. So I think that's probably pretty behavioral, that decrease.
And one other question I had related to the difference that you see between the ETI population and the non-ETI population. Did your group consider the effects of potential confounders? Because we know that not all patients are eligible for ETI based on a variety of factors that may be related to race and ethnicity that are associated with underlying mutations in CF, or potentially insurance or socioeconomic status that might also then be associated with their risk for exposure to viral infections or other precipitating factors in the community. Did you guys look at any of those potential demographics or other confounders?
Yeah, so one thing that was accounted for was, of course, within the non-ETI population, patients with that group contained patients that were taking modulator at least. So any of the more severe mutations that would have, disqualified people for some kind of a modulator therapy were not included. That kind of analysis in terms of confounders was not done to the level that you're discussing. And that's, a really interesting point, because especially within New York City and the patient population that's followed at Columbia's CF Center, it's pretty diverse. And of course, to an extent, there's going to be variables like that involved. So that's definitely something that, we're interested in and kind of digging in deeper to, and, potentially seeing if you can parse that out on the population level. Yeah. Thank you.
Sorry, I'm muted there. That was an excellent talk, but we've answered all the questions from the audience. So let's move on to our fifth talk for this session, who is Padraig Hawkins, who's going to present reduced susceptibility of mice with CF-like lung disease to SARS-CoV-2 infection.
Hello, my name is Padraig Hawkins. I'm a postdoctoral researcher in the Boucher Lab at the University of North Carolina, Chapel Hill. I'd like to thank the organizers for the opportunity to present this work. The title of my presentation is Reduced Susceptibility of Mice with CF-like Lung Disease to SARS-CoV-2 Infection. There are no relationships to disclose related to this presentation. We know that respiratory viral infections in people with CF are a common cause of morbidity. They may be associated with more severe and prolonged declines in FEV1, And in children, it has been shown that they increase the likelihood of subsequent airway colonization with bacteria. SARS-CoV-2, a respiratory virus, continues to exert an unprecedented burden of global health services and economies. Epidemiological data to date suggests that individuals with CF are in fact not at increased risk of infection with SARS-CoV-2, and indeed, with the exception of a small subset of individuals, do not suffer more severe COVID-19 disease. These findings are likely related to exceptional adherence to infection control measures that mitigate the chance of acquiring SARS-2 by individuals with CF. A seemingly contrasting finding is that airway epithelial cells from individuals with CF show increased abundance of SARS-2 entry factors, TMPRSS2 and ACE2. We felt that it was important that we attempt to further understand the biology of SARS-2 in individuals with CF. We hypothesized that individuals with CF are protected from infection with SARS-2 due to airway mucus accumulation. Using the beta-ENaC mouse as a model of CF-like lung disease, we set out firstly to quantify SARS-2 infection and to examine the tropism of SARS-2 infection compared to wild-type mice. By way of background, inoculation of mice with virus was performed in the BSL-3 of Dr. Ralph Barrick here at UNC. The virus used was the original Wuhan strain, which was manipulated using reverse genetics in order to remodel the spike and receptor binding interface to enable successful binding to the mouse ACE2 receptor. This novel mouse-adapted virus has been shown to recapitulate the key features of COVID-19 in humans. For our experiments, we chose to use a sublethal inoculum of mouse-adapted SARS-2. As we can see from these weight loss curves, this dose of 10 to the 4 PFU resulted in modest weight loss peaking at day 3 and no mortality. In this model, infection with SARS-2 was seen almost exclusively in airway club cells and alveolar type 2 cells, and there was histological evidence of acute lung injury. We chose to use the beta-ENaC mouse for these studies. This is an extensively studied model of muco-obstructive lung disease. The clinical, pathological, and physiological phenotype of this mouse bears many similarities of CF lung disease. For example, these mice show an increased percent solids of their ASL with a reduction in the height of their PCL, airway mucus obstruction, and bacterial colonization. The use of this mouse model with the novel mouse-adapted SARS-2 virus provided us with a unique opportunity to study the impact of SARS-2 infection on muco-obstructive airways diseases. With regards to our experimental design, in the BSL-3 facility, 10 to 12-week-old beta-ENaC mice and wild-type littermate controls were inoculated intranasally with 10 to the 4 PFU in 50 microliters of PBS of mouse-adapted SARS-2 virus. Necropsy and harvest of lung tissue was performed at various time points post-inoculation. Moving on to the results. Viral titer at 2 days post-infection, as shown here, was not different between wild-type and beta-ENaC mice. This was determined by plaque assay of homogenized right caudal lobes. However, beta-ENaC mice experienced significantly less weight loss than wild-type controls. Of great interest to us was the striking difference in the pattern of infection between beta-ENaC mice and control mice when examined using immunohistochemistry and RNA in situ hybridization. Here you can see immunohistochemistry performed for SARS-2 nucleocapsid protein on left lung lobes harvested at 2 days post-infection. Wild type is on the left and beta-ENaC on the right. The most striking difference is near absence of airway infection in the beta-ENaC mouse compared to the controls. Quantification of the IHC signal showed that there was no difference in whole lung or alveolar viral abundance, while there was a near 3-fold lower abundance of virus in the airways of beta-ENaC mice. These results were confirmed using RNA in situ hybridization, and these images show serial sections of the prior slides where SARS-CoV-2 spike mRNA is shown in white, its surfactant protein C signal shown in red with DAPI staining of nuclei in blue. Again, we can see in the higher magnification images here that the airways of beta-ENaC mice have far less airway infection. We next wanted to understand why beta mice airway epithelial cells are protected from SARS-2 infection. We first look at airway mucus and secretomucins, as this is a model of muco-obstructive airways. What I'm showing you here is a wild-type mouse on top and a beta mouse on the bottom at 2 days post-inoculation with SARS-2. Serial section of these left lobes were stained for mucus using ABPS, MUC5B protein, and SARS-CoV-2 nucleocapsid protein by immunohistochemistry. In order to understand the relationship of viral infection to mucus. As expected, we see increased areas of ABPS positive mucus in the beta mice that stains robustly for MUC5B protein. Airways with more abundant mucus were less likely to stain for SARS-CoV-2 nucleocapsid protein. This suggests that accumulated airway mucus in beta mice offers protection of the underlying airway epithelium to SARS-2 infection. We are breeding a line of secretomucin deficient beta mice to further validate our findings. We next wanted to search for other possible explanations as to the protection of beta mouse airway epithelial cells from SARS-2 infection. In order to further characterize these beta mice, we performed 10x single-cell RNA sequencing of whole lung suspensions and examined SARS-2 entry receptors and antiviral gene expression in beta versus wild-type mice lungs. This UMAP here on the left shows 34 clusters generated from our 10x single-cell experiments. I have selected a small number of genes to show here, and they're displayed as violin plots with beta in orange and wild-type in green. We found that there is no difference in the level of expression of ACE2 or TMPRSS2 between wild-type and beta mice. Here I'm focusing on club cells, which you can see are labeled as cluster 30. You will recall that the mouse adaptovirus infects club cells and not ciliated cells in the airway epithelium. Next, we looked at genes with antiviral activity and found a number of genes that showed much greater expression in beta compared to wild-type mice. The increased expression of these genes, namely IFITM1, MUC4, and BPIFB1 in club cells of beta mice, may offer another explanation as to the lack of SARS-2 infection in beta club cells. Having looked at the acute consequences of SARS-2 infection, we next examined for evidence of lung disease in beta or wild-type mice at later time points post-infection. We were quite surprised by our findings. The lungs of beta mice showed grossly abnormal areas of dense inflammatory infiltration by 15 days post-inoculation, and this had become more organized by 30 days post-inoculation. The H&E stained sections of mouse lungs on the right are representative images of wild-type and beta mice at 15, 30, and 60 days post-inoculation. You can clearly see the discrete areas of parenchymal consolidation. Wild-type mice showed no histological evidence of lung injury or disease after 15 days post-inoculation. We examined this lesion in more detail using immunohistochemistry and RNA in situ hybridization. These images are from wild-type and beta mice at 30 days post-inoculation. We can see in this high power of a H&E stained section of the beta mouse that there is gross distortion of the lung structure and the presence of needle-shaped crystals. There is accumulation of eosinophils as seen with IHC for major basic protein. RNA in situ hybridization revealed accumulation of great numbers of YM1 positive alternatively activated macrophages. Staining for alpha smooth muscle actin was greatly increased in beta mice in aggregated whorls suggestive of proliferating fibroblasts or myofibroblasts. These findings supported the concept that this lesion in beta mice following SARS-2 infection was a Th2 mediated process. Our next aim was to understand the origins of this Th2 mediated process. This diagram depicts some of the key players in the production of Th2 orientated cytokines. If we follow the schematic, we see that some form of epithelial injury or allergen exposure causes the release of IL-33, IL-25 or TSLP and TSLP from the epithelium. IL-33 acts via the ST2 receptor on mast cells, basal cells, ILC2s, Th2 cells, and macrophages. to promote the production of IL-13, IL-5, and IL-10. IL-33 is constitutively expressed in 82 cells of mouse lungs, meaning that it may be released upon infection and injury of 82 cells with SARS-2. From the literature, we know that increased serum IL-33 levels have been found to be independently predictive of poor outcome with SARS-CoV-2 in patients less than 70 years old. Additionally, transcriptomic analysis of BAL fluid from COVID-19 patients demonstrated upregulation of IL-33. Furthermore, infection of human primary H2 alveolus virus resulted in increased expression of IL-33. For this reason, we deemed IL-33 to be a candidate cytokine to study in more detail in beta-ENaC mice. We found from our single-cell data that 2 of 4 H2 cell clusters showed increased expression of IL-33 in beta-ENaC mice compared to wild-type mice. RNA in situ hybridization confirmed that in uninfected mice, IL-33 was predominantly expressed in H2 cells, as seen with co-localization of IL-33 with surfactant protein C, and that expression was increased in beta-ENaC mice compared to wild-type mice. There was marked increase in IL-33 expression in beta-ENaC mice at 30 days post-infection within the abnormal areas of lung parenchyma. To summarize, we have shown that in a mouse model of chronic mucus obstruction, SARS-CoV-2 causes less severe acute disease. Airway secreted mucins and accumulation of air mucus are associated with less airway epithelial infection with SARS-CoV-2. Beta-ENaC mice develop a chronic Th2-dominated organizing pneumonia type lesion following infection with SARS-CoV-2. IL-33 expression in H2 cells is elevated in beta-ENaC mice at baseline and may drive the development of chronic lung disease seen following SARS-CoV-2 infection. These studies are in their early stages, and our next steps will be to perform whole transcriptome digital spatial profiling to characterize the pathways in beta-ENaC mice that are responsible for the development of the Th2-dominated lesion after SARS-2 infection. Ongoing studies are examining a number of therapies for the chronic post-SARS-2 lung disease, including corticosteroids. We are in parallel studying the impact of other coronavirus on the beta-ENaC mouse in the acute and chronic settings. I've had a lot of support to perform these studies. I'd like to thank everybody in the Marsco Lung Institute and the Barrack Lab who have helped with these. I'd like to thank the Cystic Fibrosis Foundation for their support via a postdoctoral research fellowship award. And thank you for listening.
Thank you for that excellent talk. Let's go ahead and get to the questions from the audience. So Alejandro Pizzulo says, that was a great presentation. Did you obtain BALs for bacterial culture from the beta-ENaC mice? And do they have bacterial colonization prior to the viral infection?
Yeah, that's a great question. Thank you very much, Alejandro. We didn't, and unfortunately, you know, we'd be very much interested in getting BAL from these mice, but unfortunately to date, we haven't, or our BSL-3 hasn't had the capability to do that, you know, due to EHS protocols. They haven't come up with a protocol where we can safely get BAL out of the, or approved protocols where we can get the BAL out. What I can say is we've done RNA in situ hybridization for 16S RNA, and we've done it at later time points to be sure that, you know, what we're seeing isn't in some way related to, you know, massive amounts of bacteria in the airways, and we haven't found a significant amount of the 16S RNA based on RNA in situ hybridization.
Maybe as a follow-up to that, do you know anything about the bacterial colonization in the beta-ENaC mice, perhaps before they're taken into the BL3? Are they at their baseline state? What's their bacterial colonization in their lungs look like?
Yeah, so I mean, it's been fairly extensively studied here by others at the Marsco Lung Institute, and at baseline compared to wild type, they do tend to have more bacteria, excuse me, more bacteria, and they tend to be kind of, you know, bacteria from the oropharynx that are probably aspirated.
And a question that I had is, so you showed early on in your talk that the overall viral titers, and I believe in the one lung that you homogenated, were similar between the beta-ENaC and the wild type mice, but there was, in your immunohistochemistry, obvious differences in the amount of virus that was present in the airway cells. Is that, do you think, because the total amount of virus that's out in the alveolar tissue kind of overwhelms and makes the total amount of virus in the lung similar, or is there another reason that there's those disconnects between those two pieces?
Yeah, no, I think what you're saying is probably right, that the majority of the infection occurs in the alveolus, just, there are a lot more AT2 cells out there than there are airway epithelial cells, and I think that's what that reflects. I mean, the major finding I think we found was that, just the airway, probably related to the accumulation of mucus is protected in the beta-ENaC.
And then speaking about the lesions that you see in the beta-ENaC mice at 30 days, where there's this parenchymal damage that sounds like it's Th2 mediated, perhaps downstream of IL-33 based on your data. You suggested that maybe that was just a direct result of viral damage. Do you have any other potential cause for that? Is it just the viral damage that's producing the IL-33 potentially and then setting off this cascade? Or
yeah, we don't have that. That's what we're currently exploring, and we're hoping by using this digital spatial transcriptomic profiling that might give us some of those answers. I suppose our hypothesis is that at baseline at baseline beta-ENaC mice have a lot more, well, actually not a lot more, but the AT2 cells express a little bit more IL-33, and then they get infected with SARS-2 in their AT2 cells. And we know that, from human alveolosphere data, that SARS-2 induces IL-33 expression in AT2 cells. So we're assuming that is what happens with the beta-ENaC mouse. And then for whatever reason in the beta-ENaC mouse, maybe there's some, signaling via they're already activated macrophages that propagates the IL-33 and sustains it. And also there's the issue of, the chronic difficulty in clearing these things from the alveolus due to the impaired mucociliary clearance. So we feel that probably exacerbates things also.
I guess in part as a follow-up to that, you mentioned briefly at the end the thought about therapeutics for kind of the underlying effect on SARS-CoV-2 infection. Can you comment at all on what you think the effects of therapeutics that might improve mucociliary clearance might have, or what other types of therapeutics might be useful for intervening in the Th2 type response that seems to cause this more parenchymal lung damage?
Yeah, so with the improving mucociliary clearance, I think that may help in terms of, what I was saying, that, a lot of the lesions we're seeing later on are, accumulation of, macrophage and immune cells in the alveolus, which, perhaps there is some issue at the start of infection with difficulty in clearing dead or dying AT2 cells or macrophages that are infected, that are exacerbated by poor mucociliary clearance. So, that is something that we will hopefully pursue in trying mucolytics early in infection to see if that ameliorates the lesions. Other things we have been looking at, but there's some obstacles to doing is specifically targeting IL-33 or ST2, which is the receptor for IL-33. Because they're in the clinic already.
All right. I think this is an opportunity for potentially all of the panelists to turn their cameras on. And first of all, just to say thank you, all of you, for contributing your talks to our workshop, but also a chance for the audience to ask questions of all of the panelists and maybe related to some of the connections that we've seen between some of the talks. I can see some cameras on here. All right. Do we have any questions from the audience for the panel as a whole from today's workshop presenters? One question I had was related to the potential connection between Shreya's talk, where she's seeing the effects of IL-13 as far as initial protection from infection in some cells in their bronchial epithelial cells, and what you've seen in your talks. Do you, now that you've had a chance to see each other's, see the data presented by each of you, do you think that the model that that presented in Shreya's represents kind of this decrease in infection that you're seeing in the airways, as opposed to the process that you're seeing more at the alveolar level? I can't see if we still have everybody here.
Yeah, so I suppose, yeah, Shreya's work was very interesting. I suppose in using mouse models, you know, there are some limitations and obviously differences with using human airway cultures. And, you know, one difference in mice is that IL-33 is constitutively expressed in AT2 cells, and that is slightly different to humans, where it is constitutively expressed, you know, more in airway cells. So, you know, our hypothesis would be from our point of view is that, you know, in people with CF, that, you know, injury with the virus would actually cause upregulation of IL-33 in their damaged airways, and specifically their basal cells, you know, to induce IL-33 and goblet cell metaplasia in their airways, rather than what we are seeing in our mice.
Great. I'd like to give everybody a chance to have a minute or two if they'd like to have any closing thoughts, or otherwise. So if anybody, after having seen all these presentations, has any thoughts that they'd like to share, that'd be great. So please go ahead and do so. But if not, that's fine too. And everybody can get a few minutes of their day back. I know that for those of you on the East Coast, it's getting towards the end of the day, and it's been a great conference, but it has been 3 days on Zoom, so, or other virtual platforms. And so with that, I would like to thank all the participants in our workshop today, and everybody for contributing to a great virtual NACFC this year. Knowing that this is the last day, let's all be hopeful that we can meet in person for NACFC in 2022.